Well, thank you very much and good morning. It's a pleasure to be here today in La Jolla. I've taken the 10-minute drive from our offices to get here, and so look forward to this. This is briefly the issues that I'll touch on, and I'll move pretty rapidly since it's only a 15-minute presentation. Our company is an 8-year-old platform company at this point, developing gene and peptide therapies for the major cardiovascular and metabolic diseases. We've received substantial funding over the years from the National Institutes of Health, including a grant to conduct our first Phase 2 study in patients with congestive heart failure, and it was on that basis that we created a public-private partnership with the NIH for that study. We have a very experienced management team covering the various areas of research and development. We have a pretty extensive patent portfolio, now numbering nearly 100 of issued and pending patents. And the data from the Phase 2 clinical trial, which I'll talk about, is very impressive. We've also been actively involved in the issue of pricing and reimbursement for the past 3.5 years, actually, with the RAND Corporation, who are the recognized leaders in that space. Here's a list of our founders and key executives, and so as I said, this is really, from my perspective, an A-team in the biotech space. Here's our product pipeline, and by no means is this the extent of the products that we have, but these are the products that we are actively developing at this point. The gene therapies are all single-dose therapies. They're given once, and the effects seem to last a lifetime. We'll talk about that. And in addition, because of our extensive gene patents, we have what I like to call belt and suspenders. So for each product that you see there, we have additional very similar products that could be analogs or mutants, et cetera. So we have backups for every one of these things and many, many different products for the various cardiovascular and metabolic diseases. Now, I'll start off with our lead product, which we call RT100. It's AC6 gene transfer. And the profile of the product would lead us to believe that it would be a primary utility in people with severe congestive heart failure. The product is delivered, first of all, as part of an adenovirus 5 vector, a common cold virus, you might think of it, which has been used in about 25% of all the gene therapy trials and clinical trials in history. So there's extensive experience going back almost 30 years when I started in the field as one of the co-founders of the first gene therapy company called Vigene. It is, when used correctly, extraordinarily safe, and in our hands, both in this clinical trial that we're talking about here, but likewise in my previous company, Renova Therapeutics, the first cardiovascular gene therapy company, we've generated extensive data with more than 2,500 patient years of experience, and the adenovirus 5-based gene therapies that we've developed have a safety profile equivalent to placebo, so really extraordinary. And in terms of the congestive heart failure treatment using the AC6 or adenyl cyclase 6 gene, what we found here is that all of the extensive animal data that we have, more than 36 publications in the top journals in the world, each study, animal study being placebo-controlled and double-blind, what we found is that the gene therapy appears to be able to reverse congestive heart failure, not just functionally, but anatomically as well. So the hearts of these animals or humans that have low ejection fraction heart failure, so they're distended hearts, thin-walled, actually remodel and become normalized. The second product we're developing is a treatment for diabetes, and this is based on urocortin-2 gene. And we've been very fortunate enough to be able to license all the intellectual property surrounding all of the urocortin and stress-copin genes, proteins, and peptides. This gives us unbelievable products, some of which are already in development at Renova. But what we found here in the animal models of type 2 diabetes, and some of this work is published, most of the work we've done now is not yet published. Essentially, it appears to be a cure for type 2 diabetes. So after a certain period of weeks after treatment, and this product, as well as all the rest of our second generation gene therapies, are simple IV injection products. So a nurse can administer these products in a doctor's office. And over a period of weeks, usually 6 to 8 weeks, the animals are completely normalized. It's not that their diabetes is improved. They don't have diabetes anymore. This is extraordinary stuff. And we couldn't be any more excited about this. And so the initial preclinical studies have been published, as you see at the bottom, in JCI Insight. Now, another thing that we've sort of pioneered here in the gene therapy space is the development of peptide products as acute or chronic therapies as companions to our gene therapies. So specifically here, Stress-Copin is a product that we own. It was initially developed by Johnson & Johnson as a treatment for acute decompensated heart failure. And the results were wonderful, actually. But J&J believed that alongside the natural peptide, they wanted to develop a long-acting form, which they thought would have a very large commercial opportunity. And so they did that and had spectacular results in animal models of heart failure, and then initiated a clinical study which didn't work. And the reason it didn't work was that they used a pegylation, a galenical approach to increase the half-life of Stress-Copin, and that formulation did not work for reasons that people expert in that area would understand. However, we've been able to license all of this from Johnson & Johnson and in addition obtain all the intellectual property. And so in our hands, we're developing a long-acting form of this using a different approach. And we believe that this treatment could be a once-weekly subcutaneous self-administered injection by patients with needleless injectors, and that it would appear to have a profile of effects very similar to AC6. So it's an ideal product that people could self-medicate themselves with and potentially obviate the need for other therapies. Likewise, with our UroCort2 program in diabetes, we're developing a long-acting form of that peptide. And we believe that this might be, again, a huge commercial opportunity where patients with type 2 diabetes would be able to self-medicate themselves once a week and obviate the need for drugs or insulin. And so you can see that that would be a wonderful stepping stone or transition to the gene therapy, which is, of course, a one-time IV injection and has these effects throughout life. Well, let's see here. No, that's not us. So is that it? That's it. Okay, I'm sorry. So I moved too quickly. I could put in a plug for PSAV, but I don't know who they are. Okay, but at any rate, so the products that I've just briefly discussed give you an idea of the products that we're developing at Renova, but actually the actual products span a much larger net, which would include muscular dystrophy, pulmonary arterial hypertension, and other indications, NASH, that our products appear to be effective in animal models. And when I say effective, I mean they reverse the situation. So we've got an unusual situation of having a plethora of product opportunities at Renova. So we're very excited about the situation. I should also probably mention that we have intellectual property covering various vectors, and we view vectors as commodities. You know, I've been in this business since the very beginning in 1988. And what I've learned over time is that, first of all, you should choose vectors, as the British refer to it, as horses for courses. So there isn't one answer to any of these. It has to be fitted exactly to the product you're developing, the gene you intend to deliver, where you intend to deliver it, and most importantly, you need to be able to achieve high-yield gene transfer in that target organ. Now, a key to that are what we call methods of gene therapy, and we've actually patented these things, and it's proven very effective for us. So we use unique methods of delivery which do result in high-yield gene transfer to the target organ. That's why the products work. Because if you don't get enough gene transfer, regardless of whether you have the right gene or not, producing the right peptide or protein or not, if there's not enough gene transfer, you're not going to get enough peptide or protein produced. The product will not work. You can stand on your head. But if you get it right, it's like throwing a switch. And so that's the little pearl of wisdom, I guess, I have for today. So with the 4 minutes I have left, if there are any questions, be happy to answer them. Nobody? Okay. Oh, sir.
Thanks. Just wondering which AAV serotype you're using for RT200?
So we have a few different ones. We've taken a position now where we don't talk about the precise vector. I apologize for that. But for competitive reasons, and also given the size of the potential size of these products, obviously we do not want to help others to try to compete with us in any way. So we don't discuss that. I'm sorry. Anyone else? Well, thank you very much for your attention this morning. I appreciate the opportunity to speak here. Thank you.